Okay, thanks very much. And I would like to thank everybody from Chemaxon for the invitation. And next time I'll make a point of looking at the dress code before I accept the invitation. What we are going to be talking about today are the special challenges involved in enumerating and searching Markush structures and patents. The one difference, one of the very important differences between a patent and a journal article or experimental result is that an experimental result is hopefully reproducible. It's a fact. You saw X in the laboratory, as opposed to what's in a patent, which is to some extent predictive, prophetic. Nevertheless, patents are very important. They are essential to protecting chemical inventions. No drug is going to get on the market unless it can be patented. And there's a huge amount of information in patents that's just not available anywhere else. We have the figure here, 70%. I've seen other figures, but they all tend to cluster around between 2/3 and 3/4 of the information in patents is just not available anywhere, and you ignore patents at your peril. The volume of patent information is increasing constantly, and the percentage of of patents that are about chemistry is also very large. And when we talk about chemistry, we're not talking just about pharmaceutical chemistry. We're also talking about agricultural chemistry, pesticides, fertilizers, things like that, in addition to the non-life science chemical areas like dyes and motor oil additives and things like that. So it's a pretty high percentage. Now, the kind of information that you have, the kind of chemical information that you have in patents is shown here. We have specific compounds. And I have seen patent claims for drugs which the claim essentially reads, we claim X, and X is the name of a compound. There are Markushes, and I'll explain that Markush in the bottom of the screen in a moment. There are reactions in newer intermediates. There are patents out there on drugs, and then as the drug is scaled up from milligrams in the laboratory to kilograms for production, there are a lot of development steps, and some of those may be in patents as well. Somebody mentioned polymers, and to be sure, they're very important. Inorganics, nucleic acid, amino acid sequences, those are actually covered in a different database of ours called GeneSeq, which is also available, and I can talk about that if anybody's interested. But a typical Markush in a patent is shown on the screen on the right. We have the fixed scaffold, and then all the variable parts, the R groups. And sometimes the R groups are very simple. It's hydrogen or methyl or something like that. And sometimes my favorite R group is any pharmaceutically acceptable moiety. That narrows things down a lot. It has also kept patent attorneys busy in court for that sort of thing. Now, the information that the chemical information that we have in patents goes back as far as 1963, at least. Some people tell me actually earlier, but let's say 1963. In 1963, we didn't really have a lot of ways of representing the data. So we invented a fragmentation code system. There was Parallel Evolution. The company that was IFI Claims is now part of Wolters Kluwer. They developed the Claims IFI system. The GRIMA system was developed by a consortium of German and Swiss chemical companies. It's a very detailed fragmentation system. But basically what the fragmentation system does, and what's shown here for aniline, is it takes the different pieces of the molecule and basically says that they can be there, Sometimes they say that they must be there. So in this structure, for example, we have the benzene ring, which can be there, and it can be monosubstituted. These are some of the frag codes that you would see in the structure. And that you have an amine. One amine on an aromatic carbon. Not only that, but you'll also have another code which says that the amine must be there. So it's actually a little bit double-coded. And that must code is, we call it an essential code. They're actually used for searching in order to help improve the precision. And again, that's a little bit of a difficult concept to explain. And we're not talking about frag code, so I won't bother. Okay. The claim in the top right corner is the original Markush claim from Mr. Markush. Nora reminded me that Mr. Markush was actually a Hungarian immigrant to the United States. He was a chemical engineer, and he wanted to describe a dye based on aniline. And the claim from his patent is shown there in the top right corner. And then these are some of the fragmentation codes that are used to describe that compound, which is aniline, which is really the only specific compound that was mentioned in the patent. Okay, so the system evolved, and it became obvious in the 1980s that it was necessary to improve the precision, plus computing power became more powerful. So a couple of topological Markush searching systems were developed. A lot of this work was done at the University of Sheffield. Some of the people from there were involved with the Derwent effort to produce this, and also the effort from the Chemical Abstract Service. The Chem Abstract Service system eventually became MARPAT. Derwent, which is now part of Thomson Reuters, Derwent is the part of the company that hired me. Derwent worked with two other partners, INPI, the French Patent Office, and Questel, in order to develop the system which originally was called Markush Dark, is now called the Merged Markush Service. And it allowed you to, in a modern sort of way for the first time, draw a chemical structure and then find structures in the database which match what you're looking for. And that's really only half the question. The other half of the question is, then it allowed you to find the patents that mentioned the particular structure. Because all this is only important if you're doing a patentability search, which was my bread and butter for a long time. If you're doing a patentability search, you have a structure, you want to find the patents that mention it. And the structures in the database are just a route to that patent. So this was, the Markush Dark system was the first one. It was followed a year later by the MARPAT system. They use somewhat different approaches to do the Markush searching, but those are the first ones. All right, fast forward. Fast forward a little bit, we have, since that time, we've developed a lot of new tools. The cheminformatics community has developed a lot of new tools. I'm actually only going to mention one or two, since I suspect that you guys are probably more up on this than I am. One of them is the text mining, IBM, of course. Steve mentioned the IBM project yesterday, which is very impressive. ACD Labs, for example, has a program for generating names from chemical structures. And that's actually what drug companies used to do for an awful long time. They had chemical nomenclature specialists to do that sort of thing. I suspect they're all now with the slide rule specialists. So what we have now, all right, now before I address what's on the slide, let me also give you a little bit of another story. When I first started as a patent searcher, and this is 1980, the only way we could do it was to hook in our dumb terminal to a remote system and do the search, and everything was on paper. And we would look at the abstracts and the patents on paper and keep all of our records on paper. A couple of years later came the first system that we had in my company at the time, where you could actually do the search on your own computerized workstation as part of a network. And this is probably at a time before the word intranet was even coined. Okay, now how does that compare with today? If I want to do a Markush search today, I'm in a very similar situation to the way I used to work on paper before. I can get my results, but they're only images. And there's really not much I can do with them until the Chemaxon product comes along. This allows me to Well, first of all, it allows Thomson Reuters to take the structures in Markush's, to take the Markush structures in patents and index them so that now as a searcher I can come along and find the Markush structures in the patents and then find the patents that contain the Markush structures. So the Chemaxon system is now a way of doing that and getting the structures in a way which I can now further manipulate them in a computer. The same way that now I can do a word search in a patent and get a bunch of patents and start analyzing relationships between patents on the basis of the words that are in the patents. There are also some other companies out there. Digital Chemistry, for example, specializes in cheminformatics software and a couple of other things. MARPAT is now on SciFinder. Decrypt was some software written by some people who had been with Pfizer, and I'm told that they're no longer in business. I'm not sure if that, haven't heard that confirmed anywhere, but I'm told that they're no longer in business. Okay. So what are some of the things that the merge Markush system on Chemaxon can do? It's an easy way to establish where to best focus my efforts. I said before, if I'm a drug company, I'm not going to develop any drug unless I can patent it. So I want to make sure that I avoid the patents that are already out there. It will enable me to do a kind of white space analysis. And it will also allow me to take my combinatorial libraries and start and determine which elements of the library, which compounds in the library have already been patented, and I can't really develop much more. So are we done? Would I be here if the answer were yes? There's a lot more development that is necessary. First of all, we have the problem of indexing, curation. This is done by a lot of people, and there are a lot of patents out there, and the patent load is not getting any smaller. Just to put some numbers on that, in 2009, we covered 110,000 Markush patents, patent records, inventions, actually more patents than that, but 110,000 inventions which contained Markush structures, of which just shy of 70,000 were from pharmaceutical patents. And the other 40,000 or so, again, were agricultural patents, general other kinds of chemistry patents. This involves a lot of people. This involves a lot of people sitting and reading patents and understanding them and translating the chemical content of the patent into the MMS system, into the database. The original Markush was actually fairly small. This might be a representation of that original Markush. But the more recent, the Markushes today are huge. When I teach people how to use the old Markush system, one of the examples that I use is the patent application on losartan, a fairly standard blood pressure medicine. The Markush claim on that is 11 pages. So, and it's just a recitation of every functional group and ring that the patent attorney can think of. Here is another typical real Markush, and I'm not going to ask you to find any errors on that page. And then there are some sneaky Markushes, some fairly extensive ones, like the one given here, where basically we have what looks like a small molecule, except every single position can be any number of R groups, atoms, whatever. Okay, now again, the Markush structures are good and they're important and all that, but that's only half of what we have to worry about, because patents will talk about a structure, but only if the structure is useful. That's one of the requirements for being in a patent. Patent has to be new, useful, and obvious, and our structures also have to have some disclosed use. The way we handle that is with the abstract, and the abstracts are also searchable on the Chemaxon system. So here we see on the left, we see a structure that we might have found with a typical chemical structure search. And then on the right, we have the abstract, and the abstract is actually broken down into several different paragraphs. So you can quickly zero in on the parts of the abstract that are most relevant to you. Is this a patent about the manufacture of the compound? Is this a patent about new uses of the compound? Things like that. And the way that's represented in the database is shown here. This is actually a piece of a DWPI record. The first big paragraph in the top left, excuse me, most of those 4-character strings, letter, number, most of them relate to different structural features of the Markush, and that's the old fragmentation code system. Even though it goes back to 1963, customers still like them, and so we still produce them. Some of the codes also talk about how is the compound made, what bonds are made during the manufacture, what bonds are broken, is it an oxidation reaction, things like that. A lot of the codes that are in green, represent what it is that the compound does. It's a blood pressure medicine, it's a dye, whatever. That's the first paragraph. It also contains links to the Markush number. And that's really the key to, one of the keys to making this whole thing work, is that number where it says MCN in brown, this number represents the accession number, if you will, of the Markush structure. The rest of the paragraphs all relate to specifically disclosed embodiments of the compound. So that paragraph will be about one of the specific compounds that's either a real embodiment of the Markush or a different compound that's mentioned in the patent. Okay, and again, the different fragmentation codes are shown here, and this gives you just a very quick idea of the kind of information that's in there. And this slide shows some of the compounds, some of the specific compounds. And again, the specific compounds that may be indexed may be specific embodiments of the Markush, or they may be other compounds that are mentioned by the patent. Okay, so let's go back. I'm a patent searcher. I do my search. I find the patent that describes my structure. I need to figure out how my query matches the particular Markush that was found. And by using the Chemaxon software, that's actually fairly straightforward. I'll find my hits, my structures, and does it really match, and how does it really match? Because the attorneys and the scientists that I'm working for want to know how close a match it is. And we can determine that by using some of the special features of the Chemaxon, the Markush enumeration features that Zabalks mentioned a couple of minutes ago. The next step is to try to figure out where in the body of art, in the body of prior art, where does my compound fit? We can already do that with text analysis. What we're looking at here is not a map of Jurassic Park. It's a topological map of a real body of prior art, which is done with text. And what is conceptually possible once you have your Markush query and patents with the Markush structures is to do something similar with structures as we currently do with text. And that's one of the possible goals. It will also enable us to ask better questions. These are some of the these are some questions for the future. Can we do more sophisticated queries in order to help evaluate our compounds and the invention? Can we do this with much less extensive training? You know, I train people how to use the Markush system. It takes a half a day. Most of the other systems I train people how to use takes about an hour. Can you do it in less time? Can you get better results in less time? And this is, again, something which, if you already know the Chemaxon system, there's probably not that much extra for you to learn in order to be able to interrogate the Markush data on Chemaxon. Questions? Do we have time for questions?
Well,
Are there any mechanism for differentiating between independent and dependent product claims?
Question is there any way of distinguishing between independent claims and dependent claims? And let me anticipate one of the other questions that's being asked in the audience, and that is, what is an independent claim and what's a dependent claim? The actual scope, the legal scope of an invention is defined by the claims, and in most countries that's usually some text at the back of the patent. A typical claim might be, we claim a drug, we claim a compound as shown in Formula 1, and then a dependent claim will say something like a compound as in Formula 1, which is used to treat hypertension. A dependent claim always refers to an independent claim. If the claim does not refer to anything else, then it's an independent claim. Okay, so let me ask Ken's question again. Can you distinguish between them? And the answer is actually at this point no. We have tags that you can attach to structures which will indicate whether the structure is claimed or whether it's from an example or certain other things. But we do not distinguish between dependent and independent claims. Okay, just let me repeat the question, if nothing else, just for the camera. The first claim, the broadest claim, usually, will say we claim a compound as in one, and it has all these different R groups. And then a subsequent claim, a dependent claim, may actually claim a compound which is a specific embodiment to that. And the question is, can you distinguish between those sorts of things? And again, the answer is no. That would have to be done during the indexing process by the people who are actually reading and translating that, and we haven't done that.
thank you. Are there other questions? Yes, Pat.
Just curious as to how often you do control studies. So you say, okay, I'm going to have 3 different people index
Mm-hmm.
the same patent, and how much overlap is there between one and the other?
Okay, question there is how often do we do control sets to make sure that different people are indexing patents in the same way? That's part of the quality control that we have of the database, and it takes years to get an indexer really up and running on their own. The quality checks are pretty extensive, and I can't really address it. I've never been an indexer. I approach this whole thing from the perspective of a user rather than an indexer. But I know, and I've seen reports on our quality, and I know that there is a lot of control over quality.
Great. Thanks. Let's thanks Don again.